I have to say that this is my first time getting involved with the PD community, so I'm absolutely amazed about that, and I want to thank everyone, all of you, for being here. It's lunchtime. After all the exercise and all these hard words, I'm pretty sure that everyone is starving, and I have the honor to do the last talk before lunch. It's going to be complex, but I can promise you that I hopefully will try to do my job, trying to dissect all these complex words for you to understand what we are doing from my side of view, my passion, which is the basic research. All the speakers this morning have been trying to share with you their passions. I can tell you that my passion is research, but it's research that we can use later on to help patients, and that's what I'm hoping I can achieve someday. So let's start to think about the God. Since it's lunch coming, I'm going to be bringing a little bit of entry these plates for you to get ready for lunch. So let's start talking about the gut and the brain. And I just bring this as an example for you to try to understand what we are doing on my lab. So I think about the brain as our sun right now. You can see it there, and you will see how the gut is down in our body. So the first question that we can have most of the time is how they are connected. They are not really that close. So what is involved in that? And it seems that it's pretty much impossible that they could be connected. It's a long way from the gut to get into the brain, right? So that is tricky, but I can tell you, and hopefully through the entire presentation, I can explain a little bit to you that they are connected. And I will show you a little bit about how they are connected. So if we think about the gut, we imagine the gut like a tube. It's a closed tube, a long one, I have to say. It's one of the long organs in our body, which is impressive. Inside the tube, you will see these little fingers that are on the figure, and those fingers are involved in all the digestive processes. Now, if we take a zoom in of those little fingers, you will see that we have a lot of cells there. We have cells that are aligned through the entire fingers. We call them epithelial cells, and they help us to take all the nutrients from the food that we eat. Now, the gut has so many other components. We have nerves there, and you can see the nerves represented in our picture in green. And we also have veins that bring blood circulation into them so they can work, because they need oxygen. And the blood circulation is represented as the network that you can see in blue and red. But in addition, in the lumen of this tube, that means where the food is, we have the microbiome. The microbiome are little bugs that we always have in our intestine. They live with us. We need them, and they need us. And that's how they help us to process the food and the nutrients that we get from the food. The other component that we also have in the gut are the immune cells. And those little dots that I put there are the cells. The immune cells, I think about the immune cells as our police officers. So these police officers are the ones that travel all around our body to help us to keep everything under control. They help us to control pathogens so we don't get infections. They help us to produce something that we call antibodies so we can get protected against infections. So if you think about antibodies, the best example for me right now to bring on is COVID vaccines. And we are all told every day that we need to get the COVID vaccines so we can stimulate all these police officers so they can protect us against COVID. So that's what they are doing there every day, every minute, to help us to control pathogens. Now, what are they doing there? Why the microbiome that we have in the gut is important? And maybe some of you will be asking what could happen if we get rid of them. So that's when this term about gut dysbiosis comes into play. So gut dysbiosis is basically a disbalance of all the different bacteria species that we have in the gut. We have different flavors of bacteria there. And somehow, in a healthy condition, they are all balanced. We have a specific amounts of each of one there. But if something happens, we see a disbalance there. Some are more abundant, some disappear, some decrease in terms of amount, and that's when gut dysbiosis comes into play. The gut dysbiosis damages that intestinal epithelial layer that I showed you before. So it breaks down, and then it produces inflammation. So our police officers can sense that and say, oh, something is happening, and they bring on all their tools to defend us. And that's when pathologies are coming into the gut. Now, the thing is that we know now that the gut is not by itself in our body, and it can communicate with the brain through different pathways. You can see in the picture a lot of things happening there. I'm not going to explain them to you right now, just because not all of them are important for my presentation, but I can tell you that the epithelial cells that I showed you in the little fingers in the gut are important. We also have our police officers. They are always sitting there. They are also important to control inflammation. They produce substances there to balance everything in the gut. And the neurons in the gut are one of the ones that communicate what is happening in the gut with the brain. So we transmit lots of information from the gut into the brain through nerves, through all the substances that we are produced. Some of those substances and some of those police officers can migrate through the blood circulation from the gut into the brain. And that's how both organs can communicate. Now, there are different ways they can communicate. So since we know that there is gut-brain axis, meaning a communication between the gut and the brain, what we are doing pretty much in our lab is that we are trying to dissect all the different components in the gut, including the gut microbiome, as well as our police officers, and how they can impact brain disorders. And the first thing that I was doing in my lab was working with multiple sclerosis. For you that maybe are not familiar with multiple sclerosis, I can tell you briefly that multiple sclerosis is an autoimmune disease that affects the brain. It damages the neurons, the myelin of the neurons, and then the patients develop progressive motor dysfunction as well as other systemic symptoms. The brain decreases its size over time, and it's also a really progressive disease. But what is the reason I'm bringing multiple sclerosis here is because multiple sclerosis is one of the diseases where we are really advanced in terms of the knowledge of how the gut microbiome can impact the disease. So there are several good papers, beautiful papers, showing that depending on the composition of the bacteria that we have in the gut, we can be more susceptible or less susceptible to develop multiple sclerosis. And I have here just two of the good papers that we have so far for multiple sclerosis, showing the specific bacteria that can increase or decrease during the disease. As you can see, the names of the bacteria are even harder to pronounce. We have names that I'm not going to make you name them just because they are crazy. It's just for scientists who can decipher what they mean. But I just bring some examples of the ones that are increasing. And we know that once they increase in our gut, they produce more substances that make us more inflamed. And that damages also the brain, and that's how most of those patients are highly susceptible to develop MS or to have more progression into the disease. So MS has been helping us to really understand what is happening between gut and the brain and brain diseases. Once we started working in the gut in MS, we decided to start a few preclinical studies to validate what was happening in the gut with those cells. So for that, we have the experimental autoimmune encephalomyelitis model, which is a model that we use in mice to mimic the disease. It's not perfect, but it's one of the good tools that we have to understand what is happening during brain disorders. So there is a way that we can induce the disease in mice, and then we track them over time, and we score them. We score the symptoms in the mice. In a similar way, we assess the symptoms and the motor dysfunction in patients. And we can see how develop paralysis over time. And this model helped us to realize that there were cells in the gut, some of those police officers that were recognized in the gut microbiome and were going into the brain to help us decrease the inflammation. So by the end, we focus on these police officers that we call them plasma cells. So I think about these plasma cells as a small group of police officers that are highly skilled on just a few jobs. The main jobs that those police officers have are they express one molecule that it's important for us to identify us. It's called CD138. So by that, using that marker, we can identify where those cells are and track them to see if they get into the brain. And in terms of the specialized skills that they have, I can tell you that they produce antibodies. The antibodies are substances that we need to protect us against pathogens. There are different substances, but I just put here two of the most important antibodies. And why this is important? Most of the B cells that produce the IgA substance, we know are coming from the gut. And most of the plasma cells that are producing a different substance that it's called IgG are derived from the blood, from different organs that are not the gut. So these help us to know which ones are coming from the gut, and we use that to differentiate them. And in addition, they produce other substances that can increase inflammation, meaning damage in the tissues, or decrease inflammation, meaning helping to restore the homeostasis in the tissues. And we track them with different tools that we have in the lab to track the cells in the mice. And then we track them in the gut as well as in the brain during our MS model in mice. So I'm just showing you here now an image that we can obtain from those mice after they are almost totally paralyzed from the gut. So the images are just representing the gut. You can see some patches of yellow dots on the two first images. Those yellow dots represent cells, so each dot is one cell. And that tissue, that is gut tissue, right? And then we use the CD138 marker that I mentioned before that we use to track plasma cells to identify them in the gut. So the blue dots that are on the bottom of the images in blue, those blue dots are plasma cells. Each dot is one plasma cell. So maybe you might think, oh, they are a lot. So the answer is yes. In healthy individuals, the gut is the main source of those blue dots, right? And we know that they produce IgA. Now, when you take a look on the right panel, we have the gut staining derived from mice that had the MS model. And as you can see, if you compare the both bottom panels in blue, the healthy mice do have lots of blue dots, right? But if you take a look of the next panel, the blue dots in those mice that are sick are not that many. You can't really see that many blue dots there. And by the beginning, we were really surprised about that. Over time, we were able to demonstrate that the cells were really decreased, and the epithelial layer that I showed you in those fingers were totally damaged during EAE. Now, you might think, well, but EAE and MS, it's a brain disorder, so how come something is happening in the gut? And that was exactly the same question that we had by the beginning. And through all the research, we were able to show and develop a model that was suggesting that we saw a decrease of those blue dots in the gut because they were moving into the brain once our body sensed that there was a disease in the brain. So overall, we were able to come up with this model for MS. We know that the gut microbiome is in the gut. Once the plasma cells get in contact with that gut microbiome, they are prone to produce substances that decreases inflammation or decreases damage. At some point, they go to some reservoir tissues. They stay there and they wait until they are called for job. And if there is damage in the brain, they sense, okay, something is happening in the brain. They are calling us 911, we need to go to the brain, right? And they go into the brain. They start to producing those substances to decrease inflammation in the brain, and they help us to produce something that we call anti-inflammatory immune response. That means those cells help us to decrease the damage in the brain. Now, I'm not going to show you all the experiments that we did there, but we were able to show that once we inject those specific cells into mice that are sick, we can improve the symptoms of the patient. So the mice do better once we inject those cells there. But remember, they are derived from the gut. So we were able to say those plasma cells are originated in the gut because of the presence of the gut microbiome, and then they can travel into the brain when they sense danger. So then the question is, okay, I'm here because of Parkinson. So how this is all related with Parkinson's disease? And this is a really nice open question for me. And the reason is because there is really little evidence about how the gut microbiome can impact Parkinson's disease. And the main questions that we are trying to understand right now in the lab is, one, what is the impact of the gut microbiome in Parkinson's disease? I have to say that I'm not the only one that is trying to understand this. There are different scientists all over the world doing similar things to try to address that question. The second question is, which are the possible immune cells, meaning the possible police officers, that will be related with the gut microbiome that will impact the brain during Parkinson's disease. And we are hoping that if we get more knowledge into the gut-brain axis on Parkinson, we will have a really great window opportunity to develop new therapeutics for the disease. So I have to say it's a different perspective from what we have done on Parkinson. Usually we go and check what is happening in the brain to better understand the pathology of the disease, and we try to do something there. Our perspective is a little bit more broad. We are going down to the gut, see what is happening, and see how we can impact and benefit the disease with our science. Now, I have to thank Dr. Well, I remember Nicole, but I forgot her. Dig. Oh, my goodness. I have to thank her because she did a really great job bringing the basic knowledge of what is Parkinson's disease, right? So I'm going to skip that because she's an expert on that, so I'm pretty sure that she can do a better job that I can do that, about that. But we know that Parkinson's disease is a really complicated disorder. There are so many things in place that are involved for the developing of the disease. And we also know there are multiple systems that are involved. She was able to show that there are some genetic factors that are involved with the disease, although it's only something around 30%, right? So it's not that much. We know that there are a lot of environmental factors that can be involved in increasing susceptibility to develop Parkinson. And one thing that is interesting for me is that there is evidence that there are so many gastrointestinal dysfunction symptoms that are related with the intestine that appear and be prominent even at early stages of Parkinson's disease. And I'm happy to see that there are a lot of faces around saying, yes. I hear that from patients. We are aware of that. And that's why make us think that if we try to translate our knowledge from multiple sclerosis into Parkinson, we will be able to get more knowledge about what is happening in the gut during Parkinson. Most of the time, the gastrointestinal symptoms appear at really early stages of Parkinson. So that is telling us that maybe something is happening in the gut microbiome even before we have motor symptoms in the brain. So we ask, okay, let's start thinking about what is happening in terms of the gut and our police officers in the gut and how they relate with the microbiome and later on with the brain. I can tell you that there are a few papers, believe it or not, there is not that much, although I have a big list there, but it's not that much, related with associations between gut microbiome and Parkinson's disease. The tricky issue here is that they are not big clinical trials yet. Not all of them have done with the proper techniques to analyze all the gut microbiome in the gut. So we need to do better in terms of research for that, for Parkinson. Overall, we have been able to see that there are some specific gut microbiomes, and again, I have these crazy bacteria names here, that are decreased in Parkinson's disease patients. And we know that those bacteria are involved at helping increasing the damage in the brain. So it seems that there are things happening in the gut microbiome when you are sick with Parkinson's disease. So because of that, we decided to start a pilot study where we will recruit patients with Parkinson's disease that are receiving duodopa, and I will introduce this a little bit for you in a little bit, at Toronto Western Hospital. And we are doing this in collaboration with Dr. Alfonso Fasano. He is the clinician, although I'm not a clinician anymore, so we work together on that. And we are doing it at Toronto Western Hospital at UHN back in Toronto. So what is duodopa? Maybe some of you are familiar with this, but what is known is that advanced patients with Parkinson become resistant to the levodopa. And at some point, you increase the doses of the levodopa, and that doesn't work anymore in the patients. So on those later and advanced stages of Parkinson, one option is to move those patients into a new treatment. One of the reasons the resistance is happening is because of the damage of the intestinal movement. And patients with Parkinson have constipation, and they have issues with the absorption in the gut. So we think that those patients cannot get proper concentrations of the treatment, and that's why we implement this new treatment. It's a recently new treatment to get into the gut and provide the patient with the dopamine that they need to help improve the motor symptoms. So the way this works, as you can see in the picture, is that we implant a tube in the jejunum, pretty close to the first part of the small intestine. It's pretty close to the stomach too. It's called a J-tube. And that J-tube goes out into the skin, and we fix it there, so it gives us a daily access directly into the gut. And we inject the duodopa, which is a different form of dopamine treatment, inside the jejunum directly to help the patient to get enough concentration of the dopamine. Now, all those patients are really advanced Parkinson's disease patients. And there is nothing on them about what is happening with with the gut microbiome and how that can impact the outcomes of the disease. So we decided to start the pilot study. It's in place and running now after a few months with COVID, but I can tell you that we are starting to recruiting patients, and I'm going to show you a few preliminary results that we have there, just a few patients. But this is just to give you an idea of what we are doing and what we are trying to answer here. So what we are doing is that we are collecting Parkinson's disease samples from Parkinson's disease patients that are on the duodopa treatment. That means they do have the J-tube and they are receiving the duodopa. And we age match them with a healthy volunteer. Most of the time, the healthy volunteer is the care partner or a caregiver. They are similar ages, and they are the ones that are daily involved. Now, the other reason we do it that way is because it's ideal to have the age match volunteer coming from the same in-house living hold. And the reason is because the diet can also impact the composition of the bacteria in the intestine. So if we want to compare them, we want to make sure that we collect samples from a healthy volunteer that it's eating pretty much the same diet. And then we collect a stool sample. We collect a blood sample to analyze our police officers there. The stool sample will help us to characterize the different bacteria that we have in the gut. We have them in the gut, but they are also flushed out regularly in the stool sample. So by analyzing the bacteria in the stool sample, we have a really good window to know what is happening inside the gut. And we follow the patients in the clinic with regularly follow-ups that are done in the clinic led by Dr. Fasano. So our first goal is to characterize the intestinal microbiome composition, try to understand what is happening there when patients are treated with the duodopa. And I can show you some preliminary data about that. If you take a look in red, we have the amount of bacteria that is present in the duodenum of those patients. It's really, really low, and we are still trying to understand why is that. It could be also because we are not really getting a lot of bacteria there because the nurses have to flush and clean the tube every day. So maybe it's that clean that we don't have that much bacteria there, which is good. That's what we want, right? Now, when we analyze the feces or the stool sample coming from the Parkinson's disease patients that are in orange in the left graph, you will see that we have X amount of bacteria. But when you compare the orange bar with the blue bar, you will see that in the blue bar, which is the bacteria coming from a healthy volunteer, it's a little bit higher. So that means that those duodopa patients do have less bacteria in the intestine. Now, what does that mean when we try to analyze in terms of all the different flavors of bacteria that we have there? And that's what we try to do on the other figure, where we have the microbiome composition. So the first thing that we try to do is, let's try to dissect all the big families of bacteria that are there. And what is really interesting is that they are different between patients and healthy volunteers. In orange, you have the patients. And we see how the Bacteroidetes bacteria are super increased compared with the blue bar on healthy controls. And that has been shown before in previous studies with Parkinson's disease patients. So at least this is telling us maybe we are in a good track, and we might be able to dissect some of the most important bacteria species that are increased or decreased in those patients, and then we can correlate with which are the bacteria that can increase the amount of our police officers that will help to heal the tissues, or which ones are related with increasing amount of police officers that will damage the tissue. So we know that there is overall a less amount of bacteria in PD patients, and we know that there is a decrease in some of those bacteria. Now, when we ask what will happen with our plasma cells, meaning those special police officers that we also found in multiple sclerosis that are producing substances that help us to decrease inflammation, we have a really cool technique that helps us to identify which are the plasma cells that recognize the gut microbiome in the intestine. And that will tell us that those are the plasma cells that were originated in the gut and that are circulating in the blood, and somehow maybe they can go into the brain to have an effect into the pathogenesis of the disease. So I am showing you some of the pictures that we can get from that assay to track those plasma cells in the blood. On the top is the picture coming from a healthy volunteer, and you have dots, kind of pink-red dots. Each dot is one plasma cell that is recognizing the microbiome, meaning it's producing the IgA immunoglobulin against the microbiome. So each dot was derived from the gut and somehow is circulating in the, each dot is originated in the gut and somehow it's circulating in the blood. But when you take a look of the picture that is on the bottom, the picture on the bottom is coming from one of our Parkinson's disease patients. And you can see that the number of dots there are really decreased. So then the question is, what is happening there? I don't have an answer yet, but I promise that hopefully in 2 years, we will have a better idea of what is happening. Does that mean that the cells are going somewhere, maybe the brain? Or that means that they can't leave the gut? That means that they die and they are decreased? That, I don't know, but we have lots of questions there to address. So far we know that there is an important decrease of those plasma cells that were derived from the gut microbiome that are circulating in the blood on those Parkinson's disease patients. Now, our third goal, which I think is beautiful, is we want to understand which other components that are related with the microbiome are behaving in our cohort of patients. And we call them pretty much metabolites. The metabolites are substances that are derived from diet and the bacteria in the gut, and they can impact also the composition of the microbiome, the activation of our police officers, and they also impact neurotransmitters. And we have heard a little bit about serotonin in our talks in this morning. Serotonin, it has been shown to change in Parkinson's disease patients. The gut is one of the biggest sources of serotonin. It's not the brain. So we know that serotonin can also change with changes in gut microbiome. And one of our questions here is what is happening with serotonin and other neurotransmitters when the gut microbiome is also changing, and how that can impact neurons and function in the brain. I don't have answers for that yet because we haven't started that aim yet. But so far, I can tell you that we know that there are changes in the gut microbiome in PD patients that are receiving the duodopa specifically. We know that there is an important decrease of microbiome-specific plasma cells in those patients too. The exciting thing here is that we always go back and forth between human research and preclinical studies. So we can validate some of our basic theories and then go back into patients and try to confirm what is happening there. So we are starting some preclinical studies using some mice models that we have in place in the lab for Parkinson's disease. We can transplant the fecal microbiome from Parkinson's disease patients in those mice and see how develop the disease, how they progress with the disease. And we are hoping that will help us to narrow down some of the bacteria that might have an important impact in Parkinson, and then we can go back to patients and see if we can develop new therapeutic targets that will drive the gut microbiome composition as well as those plasma cells in the gut and will help with progression in Parkinson's disease. So with that, I'm going to finish. This is pretty much all the members in the lab that have been somehow involved in the project. We have also a new master student that is involved in the pilot study with Dr. Fasano. She's just starting, so she's not in the picture yet, but she's there working really hard to get the pilot started running. I have to thank Dr. Alfonso Fasano because he has been working with me side by side with the patients. And lastly, I have to thank all the patients as well as all the caregivers who are the ones who help us to be sure that they are in the clinic on time. Every day we need them to do the clinical follow-up, to help us to collect the samples and everything. So big thanks to all of you. And with that, I'm going to finish, and I'm open to questions. Thank you so much.